Alrighty, I think everybody is already not in the waiting room anymore. Good morning and welcome everyone. My name is Dinora Chassin and I am representing the Gulf Shellfish Institute. I want to start first by thanking you for your participation. We've had very interesting talks, conversations over the last few days, and we have gathered lots of really good information. So thank you again for being part of this workshop. Yesterday was our first time trying the breakout groups discussions, and I thought it was super useful. Heard lots of valuable information in the reporting. I did get feedback from some of you that the Google Drive documents were not working properly. I just wanted to bring this to your attention. Virtual producers and I would talk, and they recommended to open the link that he shares in the chat box in Google Chrome, Mozilla Firefox, or another browser that is not Safari. If when you click on it doesn't work, just make sure that you're copying and pasting it into your browser. So hopefully that should resolve your issue. But if not, please reach out to us and let us know that is not really working. I would like to reach out to participants from group 3 and 5 from yesterday. If you remember your group, we didn't get notes from you guys. So if you can, it would be great to get an email with a couple of those take-home messages that you guys talked about yesterday. Let's see what else. Yeah, just let us know if you have any other issues. We have compiled all the information, studies, the PDF documents that you guys shared yesterday on the chat box in another folder, Google Drive folder called Day 2. And I know Brian from Virtual Producers will be posting the link in the chat box at any point now during the meeting. Again, if the link doesn't open, remember, just copy and paste it in your browser and see if that works. And at the end, not to worry, because we will compile all this information and share it anyways after the seminar has concluded. Today's meeting is going to be very similar to yesterday. We're going to have the presentations. The only difference is that we'll have 2 instead of 3. And then we're going to have a short break. And then we'll come back and reconvene. And we're going to divide into groups and do the similar process as yesterday. Not to worry if you are not present. I will definitely make sure I go through the instructions more detailed right before we break into groups. But the purpose of this is that to get all the participants talking and giving each other a chance to express their ideas, concerns, questions, gaps, pretty much any thoughts that you have from the presentations today, and definitely discuss some of those gaps that we definitely need in order to make a point that clam restoration is good. Let's see. After that, we will bring you all back, and then we will report from each group the main themes discussed, and we will hopefully have a bit more of time today for that since we have 2 talks. And I think that's all I have for now. Please, as usual, feel free to send me a message if there are any issues you might be encountering, so we can help figure it out. And now we are going to introduce the speakers for today. Let's see. Let me change slides. So our first speaker for today is Dr. Bradley Peterson. Brad is an associate professor of marine science at the School of Marine and Atmospheric Sciences at Stony Brook University. He has a bachelor's degree in marine biology from the Florida Institute of Technology, so I guess he spent some time in Florida, and a master's degree in zoology from the University of Rhode Island, and a PhD from the Dauphin Island Sea Lab, University of South Alabama. He and his research lab focus a lot of their attention on impacts of climate change, seascape fragmentation, ecosystem manipulation, and restoration ecology. Prior to joining the faculty of Oh my goodness. So mass, I guess Stony Brook Marine Science and Atmospheric Science of Stony Brook University. He served on the faculty of Southampton College as a research scientist at the Southeast Environmental Research Center at Florida International University and oversaw the Florida Keys National Marine Sanctuary Seagrass Status and Trend Monitoring Program. Brad has published over 60 peer-reviewed articles. He has served as one of the founding principal investigators in the largest and most successful bay scallop restoration project in the US, and for the Shinnecock Bay Restoration Project, which is the largest marine ecosystem manipulation project in New York. Without any further ado, please help me welcome Dr. Bradley Peterson to talk about seagrass enhancement by clams.
Can everyone see my slides? Can you see what I'm looking at?
Yes, we can.
Okay, all right. So I'm going to talk about seagrass enhancement, and I'm going to do it primarily through an example, the Shinnecock Bay Restoration Project. And I want to let you know that I have two primary, PIs on this project with me, Chris Gobler and Mike Dole. So this is a large scale, large effort. Now, this work is done in Long Island, and although it's far away from Florida, there are many similarities. The South Shore Estuary Reserve is very similar to kind of the Indian River Lagoon in many ways. So I'm going to talk about work that's done here in Shinnecock Bay. Bill started off talking about the history of the clamming industry in Florida, and he made reference to clammers coming down from Long Island. This is a series of photos from the 70s, and here are the landings from 3 of the towns in Great South Bay, which peaked in 1976 at over 700,000 bushels. During the 70s, 2 out of every 3 hard clams consumed on the east coast of the United States came from the South Shore estuaries. The population numbers declined. There was the first brown tide event in 1985. And since then, the populations of hard clams in the South Shore have not recovered. It's considered to be recruitment limited now. So by 2010, this was kind of what Shinnecock Bay looked like. There were closures on the western part of the bay for shellfishing, and the photograph, you can see the brown tide making the water clarity poor. Now at this time, Chris Gobler and I were doing a series of experiments, and we were looking at from the inlet further and further to the west in the bay, and what we saw was that the chlorophyll numbers went up, but not only did they go up, but the particle sizes got much, much smaller, so it's a poor food resource and scatters light. And at the same time, as we moved to the west, the productivity of, experimental seagrass plots declined. And we knew what our problem was. Our problem is that these estuaries are all surrounded by homes that have aging septic systems, and we have leaky nitrogen getting into our groundwater. There was an assessment done for Shinnecock Bay, and you can see that 60% of the nitrogen going into the bay is coming through our groundwater from septic systems. So in the 70s, we had a system that was primarily heavily influenced by high filtration rates, low biomass, high light, and lots of seagrass. But by 2010, our South Shore estuaries were pretty much bloom productivity. Now the productivity was no longer on the bottom, but in the water column, shading the light, and we'd lost much of our majority of our seagrasses. And so Chris and I began talking a lot about, well, how do we drive the system back from where it is to what we would like it to be? And we know that the answer ultimately was to reduce the tap of nitrogen getting into our system, but that's a billion-dollar political question. And so we began to wrestle with, well, what could we do right now? And in the early 90s, Chris had done a series of experiments looking at brown tide and hard clams. And the bottom, what you're looking at, are two mesocosms. Both of them were filled with the brown tide, but after time, the one that had hard clams on the bottom did consume them. Now, they didn't like it, and they didn't grow very well, but they were able to eat it. And so we began wondering about the ability of clams to be able to increase our biotic filtration and manipulate the system. Well, at this point, there was a lot of science that had to be done. How far to the west will the clams survive? Is their survival going to be driven by the food particle size or by environmental parameters like DO and temperature? Will they recondition after spawning? What's the residence time of Western Shinnecock? What are the benthic predators and how bad are they in consuming them? What size clam should we use that might be beyond a predation threshold? And if we set up these, spawner sanctuaries, where's the larvae going to go? Now, this was a fundamental step for success. And I bring this up because you need to know a lot about your system if you want to be successful. So we use hard clam restoration as a means to manipulate. This wasn't, what the classic idea of restoration. We were manipulating an ecosystem. We wanted to drive nitrogen out of the water column. In 2006, by the generosity of private funding, we began working on the Shinnecock Bay Restoration Project, primarily in two areas. These were where the spawner sanctuaries. I'll be referring to Weesuck Creek and Tiana Bay, and these are the two. We began work primarily in Weesuck Creek. Now you can see that we've planted a lot of adult clams. We only used adult clams because seed clams were quickly consumed. We wanted to jumpstart this restoration by fertilization success. The thinking was if we put these clams together in high density and allow them to spawn, that we would increase the larval pool into the system. But as you can see through the years, we've put out over 3 million adult hard clams. These hard clams have been put into 64 half-acre spawner sanctuaries. These are no-take zones. We can control the activity that goes on in these areas. Our target density was 25 to 35 clams. Since each of them have been plotted, there's a subset that we go back annually to look to see what our survival is. We also look at condition indices of the clams that are present in our spawner sanctuaries. A hydrodynamic model was created, and what you're looking at is the output of that model at day 14 when those larvae would be competent. The top is where the larvae is coming from Weesuck Creek. You can see it moves to the east to the west, some over here into eastern Shinnecock Bay. And this is the output of the model from Tiana Bay, where there's more entrainment within the spawner sanctuary area, but also extends much further to the west. The arrows are indicating where the Shinnecock Inlet is. Now we also do suction sampling, and we've been looking, trying to see where the clams are ending up. The bars on the graph are indicating the differences between east and west. And what you can see is that over time, the eastern part of Shinnecock Bay has greatly increased in their density per square meter of juvenile clams. When we look at the population of those hard clams, we see that there's a big difference between east and west. And in the east, those populations are primarily smaller clams, where in the west, they're the bigger chowders. So since the inception of this restoration project, which is where the arrow is in 2012, we've seen a 700% increase in the landings of hard clams in Shinnecock. And when we look at what, the component of those landings, we see that it's a disproportionate number of little necks. And if we compare kind of the landings in Shinnecock historically, 2008 and 2018 and 2019 were the best harvests in Shinnecock Bay in over 30 years. So we have, our circumstantial evidence brings us to be convinced that our restoration activities have significantly increased the number of hard clams within our systems, primarily in eastern Shinnecock, but also in western Shinnecock. The question becomes, how has this increase then impacted Shinnecock Bay? Well, these are brown tide numbers. And so what you're looking at as we move kind of further to the east, we see this decrease in brown tide, but also through the years, this decrease in brown tide. We had a particularly wet 2016 during the month of June, which kind of helps drive brown tide. And then you see in 17, it went back down. But of note, in 2018, 2019, and 2020, there's been barely detectable levels of brown tide in western Shinnecock. When we look at the hard clam closures, here you see in 2011, 14, and 15, all of western Shinnecock was closed, 4,000 acres. In 2017 and 18, that was reduced to the farthest western extent of western Shinnecock. And in 2019 and 2020, we didn't have any shellfish closures at all. Now, we have the advantage of having a long history of the county water quality monitoring. And if we use Secchi depth, which is kind of a way of looking at transparency, it's kind of a crude method, but it gives you an idea of transparency of the water. And what you're looking at now is kind of the accumulation of the first decade before restoration in western Shinnecock. And we've kind of broken the bay up into the furthest west, central, and eastern, all of western Shinnecock. And then in June, July, and August. So if we look at Secchi depths, and now bigger Secchi depths are better, and we compare that from the years past, in kind of the central and eastern extent, we see increases in Secchi depth. When we measure chlorophyll and kind of how chlorophyll's changed over that same decade before restoration, and then in the years after restoration, we kind of see a decrease in chlorophyll levels over in the west and central. But in 2017, we created a biooptical model. And what I mean is we measured chlorophyll, we measured turbidity, and we measured the colored dissolved organic matter. And using those, we can then, look at the depth at which light is reaching the bottom. We're using 20% as kind of our cutoff. Anything less than 20% is going to not allow grass to survive there over long periods of time. And you can see that that light changes over the course of the summer, right? And so we see the June, which is the years where our brown tide are the most intense, where our small particles are biggest, and then July is different, August is different, and September our season starts to wind down. Now, if we compare our biooptical model from 2017 to 2019, so over 2 years, we're just now looking at change here. What you see is that even in those 2 years, there was an improvement of, between 1% and 5% in some locations. And those locations correlate with where our spawner sanctuaries are. Now, if we treat those sanctuaries like bathtubs, and we use the filtration rates that we've measured, we can then calculate how many days it would take to filter all the water within that bathtub, kind of still area. And what we see is that both of those areas where the spawner sanctuaries are now below what Richard Dame called, they're now within biotic control, under that 4-day threshold. But, kind of anecdotally, we've been putting out baited remote underwater video since 2014 all across the bay. And the thing that struck us was that as we went through the videos in 2019 and compared them to the videos in Western Shinnecock in 2014, there's a very dramatic difference in the clarity of the water that we can see on the screens of the video cameras. So I want to talk to you about restoration and about seagrass restoration. But remember, our goal in restoration here was by manipulating the system. So how did the seagrass respond through this ecosystem manipulation? Well, here in New York, we have eelgrass, which is more similar to your haliduli down in Florida. And so it is driven by seed, and we collect seeds, reproductive shoots, in June. We then would take those reproductive shoots and we would count them out. We would put them into bags, nest-like bags. We'd attach those to buoys, and we would put them out in grids. And the idea here is that these seeds that have been pollinated would then kind of fall out of these bags in this particular area. This was a technique that was created by Chris Pickerel at Cornell, and we began to use that here for Shinnecock. This is kind of a acre of what those buoys with the seed bags attached to would look like. And then we'd come back the following year and look at the grass that had, kind of recruited underneath where those bags were and assess their shoot count and shoot density. But 4 years ago, we changed our strategy because we found that our seed predators were quite active in July when these were out. And so we wanted to increase our seed survival. And so we hold the seeds now. We let them in the lab fall. We then take them and either hand broadcast them, or more recently, we've been using burlap bags with local sediment in them to hold the seeds in place. And by doing so, we've increased our seed and seedling survival. So here's a Google map shot of where our different buoy restoration areas are. What you can see here is the grass present in 2013, all right? And then you can see what it is in 2019. So we've had grass respond and seed portions of Western Shinnecock there it was completely absent. In fact, we've got seagrass growing almost to where the canal is in some patches. Now, if we delineate all of that seagrass, we've got over 97 acres of area that's been, seagrass has taken over and has expanded. And if we use kind of the University of Rhode Island's estimate of what it costs to restore by hand an area of seagrass, that's almost $24 million worth of restorated grass in our system. Now, this caught the attention of our governor. And so he came and visited us at Sherp, and we took him out and he put some clams into a clam sanctuary. And as a result of his visit, he then ended up putting $10 million into shellfish water quality improvement in Long Island, and has designated a series of areas where the same kind of activity is occurring on the south shore of Long Island. Now, highlights of Sherp. We've continued hard clam, that recovery has continued in Shinnecock Bay. We've seen the largest hard clam landings in over 30 years. We've got, high densities of adult and juvenile clams documented by those benthic surveys. All this evidence is kind of supporting this hypothesis that the increase in clam population was caused by these activities. We've had 5 consecutive years without dense brown tide. It's the 3rd consecutive year with no brown tide at all. And we've seen a large expanse of seagrass. Now, since we're thinking about hard clam restoration, why was SHRP so successful? Well, it was data driven. It wasn't driven by politics, wasn't driven by wishful thinking. There was a lot of data that went into assessing where, why, would it be successful? We were able to create no-take zones. We had a lot of control of the bottom ground because the Southampton Town of Trustees were deeded the bottom ground by King James in 1686. So they were able to basically create no-take zones for us. We used adult clams beyond that significant predation threshold. We've used seed clams. We tend to kind of enhance areas with seed clams, but their mortality is incredibly high. The scale of the project really mattered. It has to be very large for it to have any impact on our system. And it had to continue for a long time. We started in 2012. We didn't see really any increases in the population of hard clams until 2015. Here's a cautionary note. Before SHRP began, the Nature Conservancy bought the Blue Points property, and they established a series of spawner sanctuaries within their property. And they had years of high recruitment, and they saw that recruitment die as a function of continued brown tide. They were in the middle of Great South Bay. Their spawner sanctuaries had very little control of the water column that moved across above them. They had very large predator fields where whelks would come and decimate many of their spawner sanctuaries. And so just putting hard clams in an area isn't going to result in positive activity unless you understand your system really well. And SHRP benefited greatly by what the Nature Conservancy had done and the lessons they had learned through their activities. Now, really, we're focusing now on what possibility could your hard clam restoration activities in Florida have on seagrass? Well, they can affect and increase light availability, and that was really the primary role of what was going on for SHRP. Chuck Wall, an old graduate student of mine, did a series of experiments looking at different filter feeders, hard clams, oysters, blue mussels, and their ability to increase light and significantly increased seagrass productivity. And it didn't matter what filter feeder he used, as long as he was able to increase the turnover times to 4 days, it increased the light sufficiently to affect the seagrass productivity. So I think that that's an important idea. If you want to affect light, if you want to have biotic control, you need to be able to bring your turnover time in your area to about 4 days. But not only can they affect light, they can increase sediment nutrient availability. They can fertilize the grass. 25 years ago, when I was doing my dissertation, I did it in Port St. Joe in Florida. And I was looking at mussels and mussels' ability to fertilize thalassia. In a similar way, another one of my old grad students, John Carroll, looked at hard clams and their ability to fertilize seagrass here in New York. And what he found was that they did, but their fertilization ability, the adding of the nutrient, was much more important to seagrass that was already shaded than those that weren't. And so I think that that's an important idea, right? The ability of what this nutrient addition what role it might have for seagrasses that are already in compromised light fields. You in Florida have been using this idea of fertilizing the grass for quite some time by using your bird stakes in your restoration scar, boating scar areas. And the same idea applies here. By adding nutrient addition to the seagrass, you can enhance their productivity. But you can also, by adding this nutrient resource, have an impact on their reproductive output. Not only can you affect the reproductive output by fertilizing them, but the hard clams themselves act as seed traps. They help to trap seeds and bury seeds, at least up here in New York for eelgrass, and I imagine the same would be true for haliduli down in Florida. Lisa Jackson, another student of mine, she artificially manipulated nutrient. Then we also followed this up by changing hard clams densities. And what we found was that when we added hard clams, the reproductive shoots of the eelgrass doubled their seed output. So again, I think that this additional nutrient resource can be used by the plant, not only for their productivity, but also for their reproductive output. And so, you know, when you think about how the grasses can be affected by hard clams, there's the light and there's the nutrient. There's also the idea of the, you know, kind of the habitat change and the change in the sediment complexity and being able to trap seeds. Your grass there is different than the grass we have here. You have a suite of grasses. We only have really two, and primarily one, eelgrass. So your Syringodium and your haliduli will probably be those that will respond quickest to your hard clam restoration. Thalassia, you know, it's predominantly driven by clonal development. If you wanted to bring this in, you'd really want to bring in adult shoots that would expand because their reproductive output is just much slower than what we have up here for eelgrass. So with that, I would like to take any questions that you might have.
Wonderful presentation, Bradley. Thank you so much for sharing that with us. It's definitely inspiring and very positive. So I think we definitely need lots of good news so we can see hope for estuaries for sure. All right, so I have a couple of questions here from the audience. From Patrick Baker. We have, if you can restore both clams and seagrass, you might get a positive feedback. Refugees for, I guess it's not a question. I guess he cited some studies about refugees for infantile bivalves from brook crabs, predation in Chesapeake Bay. Let's see.
Let me just say on that point there. Yep. It's a very important point. Size again matters, right? And so the ability to manipulate the system has been demonstrated. Size is important. And, you know, we're fixated on hard clams, but if you're putting the clams in the bottom, there's a lot of other things that are also going to be, attracted to that hard clam presence. And so when you think about filter feeding, although you are only focused on hard clams, there are going to be a suite of filter feeders within that hard clam complex. And so it becomes kind of a filtration mechanism. And I think that work being done up in New Hampshire, looking at kind of the upstream, downstream removal of chlorophyll are things that would be useful for your situation.
Gotcha, perfect. So we have a question from Todd Osborne. It says, great project, Brad. I think there are clear parallels between your system and the Indian River Lagoon,
yes,
which is inspiring for the work ongoing in Florida. Did you engage commercial aquaculture in your work, such as outplanting sites, etc.?
Absolutely. So, you know, I think that in many ways, many of you from Florida may remember that when the Florida Keys marine reserve came in, the commercial fishermen were not big fans, right? And it took a series of years for them to understand that by having those reserves, it actually enhanced the fishery. We had a similar thing, right? We engaged. Now, here's one of the issues. The number of baymen had significantly declined over the years. So there were only a handful of guys that were hard clamming where we were. But we worked with them. We basically made the argument that if we create these spawner sanctuaries and the larvae go other places, right, we're basically going to be bringing product to other places where you can have it. And so now what's really interesting is that even when I go to the spawner sanctuaries to assess them, I get called, because they see people over in the water at the sanctuaries, and I'm getting called by the baymen. They want to make sure, hey, who are you? What are you doing over here? This is a no-take area, because they see the importance of this for them. And so, I think buy-in by the commercial is really important. We are only beginning to grow aquaculture for oysters in Long Island. We're pretty much behind the curve, but we're trying to get aquaculture, oyster, kind of in bag up on the thing as another way of improving water quality in Western Shinnecock.
Wonderful point. I think it's definitely taking into consideration that concept of spillover and understanding it, and definitely that close partnership with the aquaculture industry is very important. At the end, they're the ones who supply us, with the clams, and so we need to keep working together and collaborating, benefiting each other and building that good relationship for sure. We have a question from Olivia Scandell. Do you know if there was less rain drought in 2018, 2020, which also would have helped the clams and brown tide?
Yeah. So the answer is 2017. The answer is yes. 2018 and 19 and 20, the answer is no. So, if you remember the brown tide, there was that spike in 16 where we had a wet year, and it's not just wet; it's wet at a particular time. It's June, it's warming temperatures and then water runoff at that particular time, because it's dissolved inorganic and organic nitrogen, which is really driving the brown tide issue. So that's really important. And in Florida, in certain parts of Florida, you're going from dry to wet season brings a big pulse of phosphorus into certain places that, kind of totally changes the dynamic of what's growing in your water. And so those are going to be really important issues, I think, for you, depending on where you are.
All right, we have a question from Jeff Beal. What is the water residence time, flushing rate of the bay?
So, all right, you notice the inlet is over on the eastern part of the bay, right? And the residence time over in the eastern part of the day is on the order of like 15 to 20 days. When we go on the western part of the bay, we're now in a residence time, depending on how far, you go. If we just keep it as an entire bay, it's about 40 days for the water to move, from one end all the way out to the other. It's like 42 based on the modeling.
Thank you. We have a question from Ashley Smith. Were there any other changes in the watershed that happened during this time?
Excellent. No, the answer is no. There are now. So recently, since the political movement is aware that our primary nitrogen problem is from septic systems, there's been a lot of activity in coming up with improved technology. Septic systems are never designed to get rid of nitrogen. They're designed to get rid of human toxic problems. But there are technologies that can remove nitrogen from septic systems. And so the county has looked at a series of them. They've established a Clean Water Institute, and there's pilot studies out now where people are kind of upgrading their septic systems. And I think that's going to be helping us. In the state of New York, there is now coming out the Long Island Nitrogen Action Plan. And the whole point there is to try to look at our watersheds and come up with the abilities to reduce them. So from 2012 to now, there's been little progress, but there's a lot of movement. And I'm hoping that they will only improve in following years.
All right, we have a question from Mark Schramek. Does the project include seagrass monitoring in areas allowed for clam harvest? Further, does clam harvest result in adverse? I think I missed the rest of that question. Sorry, hang on.
Well, our clam harvest is primarily done by bull rakes, and bull raking will tear up our grass, and it's bad. But a good clammer doesn't want to be in the grass because it isn't where, you know, his bang for the buck is going to be along the edge, where he will get higher numbers of clams and not have to get rid of the grass. So we don't see a lot of seagrass damage by them, but we do have other things. Like we have guys who go for razor clams. What they do is they basically put an outboard motor backwards and they walk that through the grass, right? And that'll just tear up, make like a scar to get the razor clams out. So depending on the fishing activity, the kind of effect it has on the grass.
Gotcha. And I think there's a comment about that question from Bill. He says, regarding Mark's last question, it's illegal to harvest clams from seagrass beds in Florida. That doesn't mean it doesn't happen, and it's very difficult to monitor divers, but for those in boats who harvest by raking is generally not a problem.
One thing that you mentioned monitoring in the last comment, right? And I think that it's really important for your restoration to have target goals and to be able to actually measure change, right? Otherwise, you don't really know what's going on. And so we've moved to drones. And so we basically have areas where we throw up a drone and then stitch the images together. That's become so cheap that I would recommend anybody to do something like this. Now, I got started in this whole plant-animal interaction with seagrasses and filter feeders as a postdoc because of the work I did in Port St. Joe. And I was called out to the West Coast Shellfish Growers Association, because they couldn't put their product in places where there was seagrass, right? They'd put their product in, and then in 3 years, there'd be a bunch of seagrass there. And much of it was because they were trapping seeds and they were fertilizing the grass, and it would show up. And I think that, you know, these are going to be issues that are going to come up for you as well.
Let's see, a question by Judy Ott. Was there any septic or watershed restoration going at the same time? I guess you kind of went over that a little bit before, but you're welcome to, you want to elaborate any more? I think that was answered. Okay, how long will it take to see water quality improvement with septic system upgrades, by Robert Basiluth?
It's going to take quite a long time because we've got, you know, a high population density, and the issue is the price point. You know, how low can we get the price down? And usually it'll probably be mandated by when the transfers of property. So you can't buy the property unless you do the upgrade. And so it'll take time for them to all go in. But there's a lot of incentives by the state right now to try to bring that price down for those people who are interested in.
Okay, we have one question from Joel Fodry. Hi, Joel. I'm thinking of the Olin et al. Paper that show water quality changes in GSV.
Great South Bay. Yeah, so
okay.
What you're talking about is the breach that was created by Superstorm Sandy. That was in Great South Bay, not in Shinnecock Bay. That had no impact on the Shinnecock Bay Restoration Project, but it had a big impact in Great South Bay. And what was really amazing is when that breach came in, a lot of colder water, right? If I could have chosen a place to make a breach to have an impact on water quality, it would have been where it occurred. And the clams responded. The growth rate of the clams after the breach in that area were phenomenal. You know, you just pick them up and you can see that growth lip was stretched. So what you're saying is absolutely true. It absolutely occurred and is continuing to occur because the breach is still open. But that was in Great South Bay, not where we were in Shinnecock Bay. And so that had no kind of impact on what we saw as changes in our water quality. Good point, though.
He has a question. following up that, he says, can you scale the environmental changes benefits you see in estuaries via large-scale clam restoration versus inlet formation widening alteration?
Well, yeah, I mean, it's a matter of your turnover time, right? And so manipulating the amount of water and the flushing rate is going to do the same thing, and it may do it quicker than putting in hard clams and having biotic control. And so I'm not going to say that one is better than the other. I'm going to say, depending on your system and your location, if you're able to improve water movement and exchange by inlet increasing or dredging, that might be a better or quicker strategy than hard clam restoration. It depends on where you are.
All righty. Well, thank you so much. We're going to wrap up the questions now. I'm going to continue this talk in the group discussion, and we're going to move on to our next speaker. Thank you so much, Matt. Brad. No, now we're going with Matt. All righty. So our next speaker is Dr. Matt Parker. If I can get this to work. All righty. Matt is the aquaculture business specialist with University of Maryland Extension. He received a bachelor's in fishery sciences from North Carolina State University in 1997, with a master's of aquaculture in 2001 from Auburn University. He completed his PhD in environmental science from the University of Maryland, focusing on the effects of debt financing on Maryland oyster aquaculture farm profitability. Before joining University of Maryland Extension, he was an aquaculture business specialist with the North Carolina Department of Agriculture and Consumer Services from 2003 to 2010. Matt is currently the president of the U.S. Aquaculture Society, and without further ado, please help me welcome Matt to talk about nutrient credits.
Thanks, Denora. Let me get my screen going here. Okay, can you see it?
Yes, we can.
All right, thank you. So I apologize, I haven't been able to attend the sessions earlier this week. It's just been one of those weeks. So I was asked to talk about nutrient credit from clams. So welcome to my presentation, Betting It All at the Clams Casino, getting credit for nutrient removal from clams. So what the end goal of this is, you've heard and you all know how great clams are for the environment, all the benefits they have, but how can you get like some credits? So like in the Chesapeake Bay here, we have a total maximum daily load, or TMDL, which is the pollution diet for the bay that can only put so much nitrogen and phosphorus into the bay. And the idea is, if you can offset what's going in, you'll have something that kind of helps and could maybe sell a credit or generate a credit or help when you can't quite meet your goals on land before it goes into the water. So what do I know about clams? They are delicious. So then why am I here? I know a lot about oysters. I'm a member of the EPA Oyster Best Management Expert Panel. So that expert panel has helped establish nutrient credit rates for oysters in the Chesapeake Bay. And Maryland has an active nutrient trading program that involves oysters, so people can get paid for the nutrients that are removed via oysters. So generally, the idea to get a best management practice approved by the EPA or someone else is, someone has to petition the EPA for permission to form a panel to look at the topic. Then they have to put together a team. You have to send all your panel members and their CVs and why you think they'd be good to the EPA, so they can say, yeah, they're good for looking at this. You have lots of panel meetings. You have lots of research that goes on to find out exactly what's out there, what we know, what we don't know. Then you have to kind of come up with recommendations and get group consensus. It's not majority rules, it's everybody has to agree. And then you have to write a report of your findings. You present them publicly to whichever agency is going to be the ones that decide whether or not this is good or bad. Here in the Chesapeake Bay, it's the Water Quality Goal Implementation Team. And then people get to comment. They get to say how awesome you are at what you decided or how much you stink at what you decided. So then it goes back and you have to address all those comments, submit the report again. If there's significant comments again, you have to address those back and forth. And then the appropriate agency members can approve or disapprove what your recommendations are. And then if things are approved, then the best management practice is created for items in the report. And then that's where your charge stops. And it's up to any municipalities to determine if there's going to be any sort of compensation. So what I'm going to do is kind of go over what our process was for oyster BMPs in the Chesapeake Bay, so you will know kind of what you have to look forward to should you guys decide to go down this route. So to establish oyster best management practices in Chesapeake Bay, we had a nonprofit group called Oyster Recovery Partnership that is very involved with aquaculture and restoration activities in the state. They've been talking about credits from oysters for a long time. They got farmer buy-in, they got restoration people buy-in. So finally they decided enough's enough, we're going to try to get this done. So in April 2015, they petitioned or submitted a request to the EPA's Chesapeake Bay office to form this expert panel to start looking at the science and seeing what's there so we can decide if we can make a best management practice. Our first panel meeting was in September 2015. And then the panel had several charges, several things we really need to consider to make sure we could do this and to do it effectively and base it soundly on science and what was available. So one, we had to identify and define all the oyster practices that we might have a BMP for. Then we had to develop the pollutant removal crediting decision framework for oyster BMPs, like how we were going to figure it out and how everything was going to go. And finally, after we did all that, we had to propose the nitrogen and phosphorus removal effectiveness estimates for oyster practices in the that we come up with that had significant or sufficient scientific support to justify our recommendations. So our projected panel end date was August 2016. So we had high hopes, but things took much longer than we expected. So for our panel, we ended up having about 16 members. We had nonprofit members, academic members, extension members, researcher members. We had every types of members you can think of. And like I said before, the goal was to reach consensus. And with everyone discussing it, it took a while. So basically what we did was, our lead Oyster Recovery Partnership did a literature review for the available science. And they came up with hundreds of papers for everyone to go through and to kind of look to see what was there. We tried to pull out the information that was available, tried to evaluate that information for appropriateness, discussed nearly every single paper, and then we even did our own independent analysis of the data that was presented so that we could try to really back up and dig into what was going on. So why, like I said, why did that take so long? So this is a picture of the practices we decided we would look at for oysters in the Chesapeake Bay. We had private oyster aquaculture from water column production and from bottom leases. We had the public fishery, and then we had oyster reef restoration. So the interesting thing about oyster reef restoration, the idea is to get the credit, you have to remove nitrogen and phosphorus from the water, which when it comes to a harvest, that's really easy to quantify. You count the oysters or the clams or whatever that comes out. Now for oyster reef restoration, that one was a little bit different. We had to wait for a review from the EPA legal office to say whether or not that was even appropriate. So it turns out that it was. So we can count, if we can come up with the science basis of it, we can count oyster reef restoration towards our nutrient removal goals and get credits for that. I would assume that since it is already said that it is good for oysters in the bay, restoration of clams in Florida would also fall into that category, but you would have to double check. So like we have here, we've got, when it comes to aquaculture, we have hatchery produced, we have some people that get wild natural set, and then the public fishery, DNR, puts out oysters for people to harvest, and then you get the wild natural set, and the same thing for oysters and restoration. So we had a whole slew of things to look at. And this is what it came down to. We had 96 different scenarios to examine for nitrogen in tissue, nitrogen in shell, denitrification, phosphorus in tissue, phosphorus in shell, suspended sediment, enhanced nitrogen burial, enhanced phosphorus burial, for every one of these things. And as you can imagine, that was a bit overwhelming to try to put into one report. So we didn't try that because I think we'd still be waiting on that one report if we had. So we decided we would take the low-hanging fruit, tissue from aquaculture. There are all kinds of studies about how much nitrogen and phosphorus are in oyster tissue. So what we did is we took all the information, we contacted the original researchers, got their data if they would share it, and we plotted it for triploid oysters and for diploid oysters. And it was based on the nitrogen and the phosphorus in the tissue based on shell height. And then we did quantitative regression and took a conservative number because people were concerned about giving too much credit or for some reason. So they didn't want to do that. So we took a conservative estimate. So we submitted our first report to the water quality goal implementation team of the Chesapeake Bay EPA program in September 2016. And it went out for public comment. We addressed the public comment, we submitted it again, and they voted on it and approved it in December 2016. So our recommendations included nitrogen and phosphorus removal from aquacultured oyster tissue. So I've got a little chart there at the bottom that tells you based on the oyster size and if it's a diploid or if it's a triploid, how much nitrogen is in, I believe it's like a gram of dried oyster tissue. Per oyster, it's not very much. So, we had hoped we would be finished by 2016, and we delivered our first report by 2016, but that left us with 86 other scenarios to consider. And just so you have a little bit, it's hard to imagine how much nitrogen is in one little bitty oyster. So we came up with a chart based on sizes for how many pounds of nitrogen and phosphorus will be reduced from 1 million oysters harvested. So for here in Maryland and in Virginia, I believe the minimum size for oyster harvest in generally is about 3 inches. So for 1 million 3-inch oysters, diploid oysters, there's 198 pounds of nitrogen reduced. And for triploid oysters, there's 287 pounds of nitrogen reduced. And it was based on sizes. So the way that I'll get to in a minute. So like there's a default estimate, which is the 3-inch size limit. If you keep awesome records, which we know everybody does, then you can go in and say, well, I sold 1 million 2.5-inch oysters, so I get 110 pounds of nitrogen, and I also sold 1 million 4-inch oysters, so I get 331 pounds of that one, and you could break it down with some extra work. So what have we been doing since December 2016? We've been trying to look at the science for everything else. So we've looked at the nitrogen and phosphorus in shell, and there's not near enough data right now for us to feel comfortable making that recommendation. We have some very smart people that work in aquatic geochemistry on the panel. So we've been looking at denitrification rates. We've been looking at sedimentation rates on how that can be reduced. We've been looking at what's been going on with the public fishery and how to credit that since it's not aquaculture. And they've also been looking at restoration activities since the EPA said that that would count towards nutrient removal. So the Oyster Recovery Partnership is still our lead group, and they are currently writing our second report that will primarily focus on denitrification. And once they get that completed, they will send it to the water quality goal implementation team, present our findings there, and then it goes for public comment. So now what about compensation? Your expert panel only determines how much nitrogen and phosphorus or whatever you're looking at is in the oyster or the clam. That's all they do. It is up to individual municipalities such as the state, the county, the city, or whoever to determine if and how compensation will be implemented. Here in Maryland, we have a water quality trading program. It was actually started back in 2010. Everyone thought it was going to be fantastic. Sewer systems needed upgrades, wastewater treatment systems needed upgrades. We were going to sell all these credits. And then the governor went and gave everyone money to upgrade all their systems and wiped out the market. So that didn't really work out so well for a while. But since then, they've been revamping it and trying to get it done. And it's been moved, everything's over in the Department of the Environment now. And they have a water quality trading program. You can go online, you can download the spreadsheet to see what credits are available, where they're from, whether they're from wastewater treatment, whether they're from traditional ag producers, or from aquaculture, from oysters. So far, we've actually had a couple of shellfish trades. There are two types of market. One is a voluntary market. The Baltimore Convention Center purchased 4 nitrogen credits in May 2020 for $1,600 from a farm. And the voluntary market is like when you go buy an airline ticket, if they ask you if you'd like to pay extra to offset some carbon that the airplane does. So that's, you voluntarily give extra money to do this. And I believe the Baltimore Convention Center is offering this type of service to people that rent the convention center if they want to offset nitrogen and help the bay, those types of things. The other market is the regulatory market, where you're trying to meet a water quality reduction goal. And our other trade was in Anne Arundel County, which is where Annapolis is. The county purchased 107 pounds of nitrogen and 12 pounds of phosphorus in June 2020 for almost $5,000 to offset runoff from impervious surface. Those are the only 2 trades we've had. We haven't had anything since. And it's difficult for producers to get in on this just because they have to keep up with the paperwork. There's 2 ways you can do it. You can do it all yourself, or you can pay an aggregator. The voluntary market went through an aggregator. The regulatory market trade we had was between the farm and the county. So it can be done both ways. So that's generally the overview. Be glad to take any questions that might have come up or you might have on kind of the process of what goes on. And I'd just like to show that I do my part to remove nitrogen from the bay as well. It's always nice to do on a boat out by the water. So that, like I said, I'd be glad to take any questions that you might have.
Thank you, Matt. That was great. What a great overview, and how much, how much you guys have had to go through in order to get to where you are, and how much there's still to go.
Well, and like Bradley said in his presentation, it takes a lot of research. You got to know what's going on, and you got to dig into it. And if you got to back it up with science. And if it's not, you can't just say, well, I think this is good. You have to say why, because you have a bunch of scientists that are evaluating it.
Absolutely. And I think it's wonderful to see the whole process of how much it takes to get to there and then go through all the science that's there, and that also in itself helps identify what are some of those key questions that we still need to find out. And I think that's like another part of this workshop is to understand that from both like how do we support restoration of clams, but also like if there is intention to use nutrient credits, how do we get there, and what do we need to know? So this has been a great example, and thank you for sharing that with us.
One of the key things is you got to have someone to spearhead the effort, and that could be a university maybe, or like the Gulf Shellfish Institute could say, we think this is a really good idea, we want to move forward with it, let's put our stuff together and submit it to whoever you need to submit to for what you need to do in Florida, you know. And then the Oyster Recovery Partnership had a staff member. That's pretty much all they did for a couple of years, was work on this and write this and work with our panel chairman and those things. So.
Absolutely, yeah, and I think that's definitely a great point. I think one thing, however, that we have seen over the course of the last few days is that compared to oysters, we don't know as much from the clam side. So that's definitely one first step that we need to do to filling all these things that you need in order to make that point. So we have one question from Gary Rollerson. Yeah, so does anyone, or I guess he asked first about anyone having links or info for Chesapeake Bay oyster nutrient criteria. And he just posted something, so I'm not sure if that's like the best thing, but do you have any recommendations, Matt?
Let me try to read his links for about the, well, yeah, what you can do is you can download the report that the group had. And I believe I had it on one of my slides. The one, yeah, it was at where I had the small table at the bottom. I had the PDF link for where the report was.
Okay, Matt, if you don't mind sharing that presentation with us, we'll definitely upload it into the folder and download that report and upload it as well into the folder so people can have it available, if that's okay.
Yeah, I sent it to your virtual producer the other day, so it should
Wonderful.
be there. And you can always Google Maryland nutrient reduction EPA, something, you know, something like that to get it.
Okay, yeah, he just messaged me. He said that we have it available. We have another question. Matt, do you have hope that nutrient credit purchases for shellfish will increase in the future? And what are some things, in your opinion, that could increase the skids to help that out?
I truly hope that more oyster farmers or anyone else are able to sell these credits. You know, my dissertation, I included a part on looking at whether or not credits would help make a farm more profitable. And if you're in the farming business of aquaculture, you know, you're in the food production, usually, you know, that type of thing. The credits are like gravy. You know, it's just extra money to help you in those hard times. You know, you can do some other things with it. So yeah, I really do hope that people will purchase more credits. And I think one of the things might be is, well, actually I had an email with the person that heads up the program, and he said he thinks we need more aggregators to kind of bundle credits together. So if a farm, say they generate 100 credits, but the wastewater treatment plant needs 1,000 credits, you know, do they want to deal with 10 different farmers and all the paperwork 10 times, or they just want to deal with the aggregator one time and bundle them together and make those big things? So that we have an aggregator here in the state that is doing those things. But if you think everything's set to go and the governor gives everyone money to upgrade everything, your market disappears. So maybe I don't want to necessarily see more people in my backyard, but if we have more people and they need to upgrade the septic systems or the septic systems for the county or the wastewater treatment systems, those types of things, you know, maybe that could drive more demand. But yeah, we've got credits just sitting there. They expire, I believe, after a year. So people are going to generate new credits. So I want people to buy them. I don't know how to make a wastewater treatment plant or a county buy a credit from an oyster farm. If I did, I'd be an aggregator and not a extension person. So.
Gotcha. I think let's see. We have a couple more questions. If you still have some more minutes.
Yeah,
Matt.
I can have a little bit more time.
Okay. All right. Sorry. Are there any carbon credits for, I guess, SAV, submerged aquatic vegetation, by Robert Vasilou?
I have no idea.
Okay.
We do not have carbon credits for SAV in Maryland or the Chesapeake Bay that I know. There might be some somewhere else, but I personally am not aware of any.
Gotcha. I apologize for that. There's some
no problem.
passing by. Let's see what else we have. A message from Thomas Aronkerson. Do you have an indication of what scale of a market will be required to make a bay or sub-bay wide difference in water quality?
I do not. Chesapeake Bay is a fairly large body of water that has a lot of water going through it. And I proposed one time that maybe we just need to put a bunch of oysters out in one river and then put another and not have them on another one and see if there's any difference. But it's really hard to tell. I don't know what scale it might be, what you might need to be. I do know that, I'm not available to stick around all day, but one of my colleagues, Dr. Suzanne Bricker from NOAA, who is also on the expert panel, is in the group. And I think she'll probably be in one of the breakout rooms. She might be able to answer that a little bit better because she's done work with nutrient removal in other parts of the country and in other countries as well.
Wonderful. We look forward to that. We have a follow-up question from Josh Patterson from that previous question on the credits. Understood. Thanks. To follow up, wastewater plants, etc., are currently purchasing credits from elsewhere?
I don't know where they might be purchasing credits from, or if the money that they were given, they were able to upgrade their system so that they did not need to purchase any credits. So that could be one aspect. Or the idea of a credit is because it would be cheaper to purchase the credit from the oyster farm than to upgrade your system. It's much cheaper to buy a credit than to expand a wastewater treatment plant or something like that. So that's the idea behind it. But if they were given money that didn't cost them free money from the state, taxpayer money or whatever to help upgrade it, then they have no desire to go out and buy a credit. So we'll just kind of have to see how it goes. And they might be doing other things such as, stormwater retention ponds, or they might be getting them from ag, if ag is traditional ag, if they're doing what they need to. So they could be getting it from some other places.
Gotcha. We have 2 more questions. Is that okay?
Yeah.
Okay. So we have a question from Sydney Williams. Thanks, Matt. Great overview. Is there an enforcement aspect to the regulatory framework? If not, is that in the works?
So here in Maryland, when you harvest from an oyster farm, we have to submit monthly harvest reports to the Maryland Department of Natural Resources. And they are the ones that validate the credit per se, or for lack of a better word, based on those harvest reports. So if you're saying, I harvested 18 billion oysters, the Department of Environment is going to say, wait a minute, that seems a little high. Let's check with DNR. Do you have the paperwork to show that you harvested 18 billion oysters? So there is they do provide that check for that. So that's how we do it in Maryland. We have I don't know how they might do it for restoration. I do know that for when they're talking about denitrification, there's some things put in there. You have to go in and measure it every once in a while if you don't want to do a low default measure and then present your findings, which measuring denitrification is fairly expensive from what I understand. So yeah, some of those aren't quite worked out, but for the oyster credit that we do have, it's verified through monthly harvest reports that are submitted to the Maryland Department of Natural Resources.
Gotcha. And let's see, one more question, or we might have 2 more. You stop me when you need to.
Okay.
A comparable is wetland mitigation banking, where mitigation is provided on larger regionally connected sites. Do you forecast other ecological benefits beyond nutrient removal with tracking nutrient credits, or is it primarily the regulatory benefit?
I think the biggest driver for it was the regulatory benefit, but I know some of my colleagues, Jeff Cornwell and Lisa Kellogg, have done some work on all the associated critters that come along when you look at restored reefs, and when you restore the reefs and put more oysters or clams or whatever in the water, there's a lot of other things that come along. And there's some good video out there of increased fish densities around oyster reefs and oyster cages and those things. And sport fishing, especially in Florida, is a huge money maker, and they spend a lot of money to go catch fish. So, if you can do things that increase the fish in the aquatic animal population, those are other ecological benefits that could come about with increased clams and oysters and that kind of thing.
And one more question we have from Bill Arnold. I hope I didn't miss this. How much nitrogen does it take to make a credit?
Well, it's based on the amount of nitrogen in the tissue of the oyster right now. So the credits are done in pounds. So, a million 3-inch diploid oysters results in 198 pounds of nitrogen. So.
Alrighty, excellent. Well, I think that wraps up the session for now. Thank you so much, Matt, for your time and for, that definitely great presentation to let us know all that we need to do and look into.
And like I said, I can't stick around for the breakout rooms. I have another meeting, but if you have some questions that come up, feel free to shoot me an email and I can get to them in the next week or so.
Absolutely. Thank you so much, Matt, for your time. We really appreciate it. Thank you to both speakers for joining us today. And we're going to take a break now, a 10-minute break. And it is 10:17. So we'll reconvene at 10:27. And then we will break into the group discussions. And I will go a little bit more in detail about the instructions for that. So we'll be back. Alrighty, and we are back after a 10-minute break. Now we're going to move into the small group discussions. So this is going to work similarly to yesterday, and if you were not present, that's totally fine. I'm just going to go over very quickly. So the groups are going to be chosen randomly. There will be about equally number of people per group. Please try to discuss both of the themes that we discussed today, the topics, for about at least 10 minutes. And of course, any great ideas or questions, gaps that flows, go ahead and choose one person to take notes of the main points of the discussion. We are going to be sharing a Google document. Brian from Virtual Producers, he's going to jump into each of our groups and adding that link there. Remember, if it doesn't quite work, copy and paste it into Google Chrome or Firefox or Mozilla Firefox. I think he recommended, and that should hopefully work. Remember to open the option of Google Doc, so you can actually type and everything will be saved. Let's see. I think that's all the instructions that we have for now. I think, Brian, if that's okay with you, perhaps you can, after you distribute the documents, maybe jump in one more time into each group just to make sure that they're not having any issues accessing that document. And I think without any further ado, we'll split you guys up and then we'll bring you back at the end and we'll just have designate yourselves a leader who is going to report back on the main topics discussed. So see you in a little bit.
Hi, Jesse and Angela. Is everything all right? Jesse, Angela, are you able to speak? You are muted.
Sorry. Yeah, I had to X out and then come back in on a different computer so I could actually use video and audio. I was in breakout room number 3.
Perfect. I'll send you back in. Thank you. And Angela, is everything all right? Hi, Tyler. Is everything all right?
I got disconnected and don't know what happened.
Oh, no worries. Do you remember what breakout room you were in?
I think I was in one.
In one with Ashley Smith, Barry Hurt, Patrick Baker, and Vincent?
Sounds right.
Okay. No worries. Do you
Thank
remember
you.
any other attendees that you might have that you could identify? Hi, Jennifer. Is everything all right? Jennifer, can you hear me?
Sorry about that. Yeah, I was having some technical difficulties on my end, so I got kicked out of the breakout room that I was originally assigned to.
No worries. Do you remember what room that was?
I believe it was room 1.
Room 1 with Ashley, Barry, Patrick, and Tyler?
Yes.
Vincent? Perfect. I'll send you there.
All righty, we are coming back. We hope you had enough time to discuss our topic. We're having definitely great group discussions. I was caught in the middle of saying something, but that was wonderful. I have really enjoyed that. So I think we have everybody back now, and I would like to now call on those leaders or at least those people who for each group took notes to come forward and give us your a summary of the main points that you discussed. If we have any volunteers from group 1, please.
Hey, it's me, Vincent, again. So we are group 1. Okay. So yeah, I can go ahead and report out.
Sure.
I'll go over Bradley Peterson's talk. I think that's generally how I've got this organized. And one thing that we learned and was discussed is that in the Florida clam farming community, in general, is advocating and in support of not only clam restoration, but this concept of a spawner sanctuary. One problem that clam farmers have, and the person that we've had in our group, Barry, is in the Charlotte Harbor area, but so on the West Coast, many of you know that red tide will shut down clam aquaculture restorations, even though the clams can survive those pretty well, especially the larger ones. But that's been, coupled with COVID has been incredibly impactful economically. And I'll get into, where the sentiment might be about nutrient creating credits in just a bit. But one thing that was mentioned is that there's not much wild clams out around some of these lease areas, and really not much wild set. Historically, and I'm speaking, I think we're using the Charlotte Harbor area as an example. Historically, there were Campachensis and Sunray Venus clams, but they're no longer there as far as wild populations. There's a sentiment expressed that would think that in Florida, there are definitely opportunities for coupling seagrass and bivalve restoration. There are seagrass mitigation banks, I think. I don't know if that's the right term to use, but certainly permitting, like say for docks and waterward structures, is very strict and intensive to minimize seagrass impacts, and maybe there's some potential there for maybe feeding that back into restoration. So these dual approaches are I think are going to be important. Another thing that was brought up was we've had a significant invasive mussel, the green mussel on the West Coast, and it's not known what those impacts might be with regards to, in this case, maybe I think what we were talking about is improving water quality. And also there was a question about predation. What was predation like in Shinnecock Bay from Brad Peterson's talk? Because that's definitely a big problem for clam aquaculture in Florida. And my guess is that part of that is seasonality as well, but I'm not sure. I'm only sort of speculating on that. And getting into those economic impacts with regards to nutrient trading credits, again, it seems like there is support from the clam industry, from clam farmers, even if the credits might not amount to a large sum, but as the clam farmers are generally small scale, maybe one guy or one or two guys, often a small benefit can be very impactful for those farmers. So I think there's definitely interest there. And because with the water quality issues with red tide and then COVID, there has, and seeing water quality decline in say places like Charlotte Harbor, they've had farmers leaving the industry, and so every little bit helps, I think, was the kind of the sentiment expressed there. And I think that for the most, there's a few more things in there. I don't want to take up too much time, but yeah, I think that covers it in general.
Perfect. Wonderful, Vincent. Thank you for sharing that with us. Now we're going to jump into group number 2. I think that's the one that I was, Lore.
All right, thank you. We had a really good talk. Everyone in our group really enjoyed Brad and Matt's talk, which served as a really good jumping off point for talking about just how to do clam restoration in our area. I think one thing that we kept going back to is that we're dealing with the differences in big scale. You know, research in our estuary is really specific and it's hard to translate from other areas, i.e. taking the work that was done in New York and translating it down here to Tampa Bay and other parts of the IRL. So I think more small scale research that is done in these areas is really necessary to be able to tease apart the drivers of what needs to happen for restoration. We had Dennis Hansack in our group, so we really went down the seagrass restoration rabbit hole, which was very interesting. And one thing that was noted was that at least in this area, seagrass restoration can only be done with shoots, not seeds, which makes it really difficult to do experiments due to the resources of just having the shoots as well as transplanting them out. And so there really needs to be a lot more basic research on seagrass restoration and the best way to pair seagrass with clam restoration. And it seems like there is avenue for a lot of different master's, PhD research. So I think that's wonderful. Another thing along the lines of seagrass restoration is we may have underestimated the role of sediment in seagrass restoration. And so obviously to restore seagrass, aka restore clams, you really need to fundamentally figure out what the sediment's role is in this. And so that seagrass status as a function of sediment is really important to get a hold of. And then the overarching conversation, I think, for probably most of these breakout sessions is that we really need to deal with the water quality, like the nutrients, so septic tanks, and just this nutrient loading that we have in these watersheds prior to restoration with clams. Just putting clams out there isn't necessarily going to take care of the problem. We need to begin at the bottom. And once we are able to figure that out, once we get our clams out, there seems to be a lack of research on how clam restoration changes the habitat structure and function over time. So maybe more chronosequence work would definitely help drive our understanding of the restoration of clams in this area as well as Tampa Bay. So thank you.
Wonderful, wonderful synopsis. Thank you. Now we're going to call on representative from group 3. Please speak up.
Michael, do you want to jump in, or did you want to jump in on this?
Yeah, I didn't know if we're group 3 or not. So what we were talking about in our group was pretty much a lot of it on soils and what sort of the optimum soils of growing these clams are. And I think Robert was doing a lot of that kind of work to provide different growers. And the majority of his work was sort of up north, but he's moving down south in Florida to figure that out. And then we talked about some of the Banana River issues out there and how there's a lack of bivalves. So there are efforts with the Shuck and Share program, I think, to restore oysters in that area. And then we talked about some of those suitability models, and it would be interesting to see if those suitability models matched up with places that had historic clam populations to see if they could still support those populations today. And if anybody wants to add anything.
Anyone else from group 3 want to add any last things before I switch to group 4? I hear an open mic. Come on. All righty, let's move to group 4. Please, Representative, speak up.
Hi, Denora, I think that's me, Gary Rollerson. Hello, everybody.
Okay, perfect.
We had some good, robust discussion. Thinking about the nutrient removal criteria. I'm from the Tampa Bay Estuary Program, and it's something that we're very focused on right now. Recognizing that this is a long-term process, understanding what, agreeing with everything that's been said going on. We need to create some sort of longer-term planning, research, policymaking process, but you got to start with what's known. That Chesapeake Bay presentation was really helpful, and I want to try to figure out how that can apply to Florida, the work that they've done, the work that others have done, and be able to bring this to us so we're not reinventing the wheel like we're always not supposed to do. And that recognizing that significant legwork is going to require communication with DEP or EPA. And again, it's not going to be something that's quick and easy. It's something that's going to be fairly long term. And posing the question, is there any current statewide numeric nutrient criteria discussion along these lines? And there's been, some things that were bubbling up in our conversation. We couldn't put our finger really on anything that was significantly happening in now the recent past. But again, thinking about that total maximum daily load, best management practices process, of course the standard names up at DEP, like Julie Espy and some others that need to be brought into a conversation like this. But also, since I think we were discussing that probably, this is from the conversation that I think occurred yesterday and again today, likely we're going to require harvesting for the most significant nutrient removal impacts, and for Florida that also means Department of Agriculture involvement, because they control the clam and the shellfish leases. So bringing them on board sooner rather than later to try to help that out. Recognizing that this will probably be a small piece of the puzzle in our highly populated state, but it's an important piece. It's, I always use that analogy of eating the elephant one bite at a time. You've got all of these different things in place, whether they are septic system removal or advanced wastewater treatment, or removal of fertilizer from lawns, or shellfish harvesting. These are different parts that can be utilized strategically to help the process. Making sure that folks like the National Estuary Programs, the research reserves, the state aquatic preserves, are involved along the step of the way, because these place-based organizations can put some of that information down on the ground as well. And we have in Tampa Bay, we have what's called the Nitrogen Management Consortium, and this is a group of 50 plus agencies and private entities around Tampa Bay that all have permitted responsibilities for nitrogen discharges into the bay, and they work together as a group and have been working together as a group since the mid-90s to ratchet down the nitrogen, and we are at a fairly fine scale where we're looking for potentially projects like this that could remove 1% or 2% of nitrogen from one of our from one of our segments. And so something like shellfish enhancement restoration could be a part of that. And we need some more, obviously, again, comprehensive assessment of how these will impact the seagrasses as well as other habitats. And for me, it's thinking about it, it's not necessarily going to be always purely the nutrient removal that's part of that seagrass restoration component attached. It's also going to be the wave attenuation that these beds can create. They're stopping the washing out of the seagrass beds while they're trying to regenerate, and I think that's a pretty important possibility for this as well. And I'll stop there and ask if anybody else from the group has anything to say, add on.
Group 4, great job, Gary. No, I have nothing else to add. Thanks.
Wonderful, wonderful. Now we're gonna move on to Group 5.
Hello, I was with Group 5. So we talked a lot about some of the different topics that came up during these presentations. The nitrogen credit system sounds like a really great idea. It sounds complex, a lot of dependencies and some work. So maybe we need somebody regionally in different parts of the state or across the country to spearhead this effort along with other restoration efforts. That sounds like a great way to move forward. Talking about the clam and seagrass presentation, their interaction and feedback sounds promising in terms of the clams increasing the filtration and light availability, increasing nutrients for seagrass, and possibly improving seagrass reproduction. And this could also in turn bring around other organisms, including other filter feeders. So a big kind of ecosystem enhancement. We were still unclear about certain things about how the brown tide interacted with this experiment and how this may also interplay with the Indian River Lagoon. On the East Coast, on the West Coast of Florida, we're not sure if we have that brown tide, but we do have the red tide, and how those interactions all interplay. We talked about concerns for sea level rise with increased water depth and temperature and climate change in terms of clam restoration. Clams could increase water clarity that may help mitigate some of these changes. A little bit of sea level rise also affects properties around these hardened shoreline areas, more so than shorelines. And so maybe this is another motivation for restoration of clams. We talked at the end a lot about soil mapping and how these maps may help identify areas for restoration. If we could get subaqueous soil well mapped, sampled, and tested. We talked with Greg Taylor, USDA, who's working on this effort with custom coring rigging and refrigerated sample storage. Currently, they're doing this in the Banana River, and we'd like to expand this over to the west coast of Florida as well. And getting the subaqueous soils mapped will help with tying it into seagrass presence, maybe perhaps species, as well as incorporating into the hard clam habitat suitability mapping product. So I think that's about it. If anybody else has anything from our group, please feel free to jump in. Thank you.
All right, that was great. I think we only have 5 groups. Do we have 6 groups? Anybody from group 6? I think we only have 6 groups. Okay, now a representative from group 6, please.
Hey, Josh, I'll go ahead and take this, but I might need some backup. I might have missed the first 1 or 2 things that we discussed. I started taking notes late, but interesting comment, Melinda, on the real quick on the USGS core samples. I think in doing some of the Banana River Lagoon around NASA, they actually found spodec layer from when those areas were maybe pine flatwoods, and now they're lagoon. So some interesting history and some effects from that in the lagoon. But we were lucky to have Brad in our group, and but selfishly, I probably talked about the Indian River Lagoon most, and there's some real correlations between the work that Brad did with that. First off, in 1994, Florida had the net ban on gill netting for fish, and I think for the IRL, Indian River Lagoon IRL, that was the first nail in the coffin in terms of seagrasses, because the clams had a high harvest of 1994 and pretty much depleted by 2012, and that's when we started seeing some of the weird blooms, and actually saw a weird bloom and a really bad one in 2016 where we had fish kills. And so there's these unintended consequences from regulation, and so you got to make sure regulation's sound, and we're still seeing that now with the work UF Whitney Labs is doing with their super clams, I'll call them, Josh, where they're sometimes regulated, can't figure out which agency regulates their clam sets, whether it's Department of Ag and Consumer Services, or whether it's FWC, and so that may be a little problematic. And to date, in the Indian River Lagoon, UF Whitney's put out 3 million clams, and so that's encouraging, and hopefully we'll continue to see more of that. One of the problems I think that they and others have seen is protection from harvesting. So we talked about protective measures. Do you need sanctuary areas, or what do you need to do to make sure these areas are protected? Just as Brad noted in his presentation, we discussed nutrient inputs, and the Indian River Lagoon Council is doing a good job through their implementation of the National Estuary Program, their management of that, working with municipalities, agencies up and down the lagoon. And certainly septic is a big culprit in Brevard County, I think at one time there were 78,000 plus or minus septic systems in the IRL watershed. So that was a big one. And the one thing we definitely see is typically, at least historically, the lagoon clears up in the winter months, the dry season here, but as soon as the rainfall starts, as soon as you turn that water on, everything upslope and including groundwater, is being pushed into the lagoon. And so that's when we get these harmful algal blooms. And that's why Suganda is going to be my new best friend, by the way. She works with HABs over in St. Pete, so that's good stuff. But the blooms we see seems like a lot higher density maybe than what Brad saw in his work. And so I know from a presentation the other day with Smithsonian Marine Labs that when we had the brown bloom here in 2016, cell concentrations were so high that clams shut down. They stopped filtering. And I suspect they can't do that very long before they die. So in contrast to what Brad did with clams, I think for like this area of the IRL, we definitely have to improve water quality to get that tipping point back to where clams can even survive, and then take that holistic approach with clams and maybe get seagrasses back and the whole system back. And then one more note, Brad brought up acidic sediments, which I haven't really thought about too much, but with the organic matter buildup, it erodes oyster shells. And so it does make it hard sometimes for survival. And we have not looked at that. I don't know if anybody's really looking at that, but that is an interesting note, because certainly where we have high organics in the sediment, also you get nutrient fluxes back into the water column. FIT has been doing some work on that. So great discussion, and I don't know if anybody has anything else to add on that.
Jeff, I think that's really good. I would just also point out that we had Jose Nunez in our group, who's with Todd Osborne at UF, working on the IRL clam restoration. So we didn't even touch on the nutrient trading credits, which was a super interesting aspect, but it seemed like between he and Brad, there was a really valuable exchange of information going on. So, yeah, we focus mostly on the clam restoration specifically, and seagrass and nutrients.
Wonderful. This just reminded me, Angela, I don't know if you wanted to mention a little bit of your work with Tom on regards on those topics, or maybe on your proposed work, or what you would like to do on that. I mean, we have time to discuss that if you want to share a little bit of that with the group.
Oh, no, no, I'm sorry, Denora. I was spaced out for a second looking at something else. I had a phone call.
Not a problem.
So I didn't hear what you said.
Yeah, no, we were just discussing nutrient credits, and we have also touched on the topic of like seagrass mitigation, all that. And it just reminded me a bit of that work I think you were proposing with Tom on doing on that regards. I don't know if you wanted to share anything with the group on that.
Oh, well, no, so there's nothing funded yet, but it's definitely been an interest throughout, obviously, as we've been talking about all day, and interest throughout the industry to develop a roadmap for Florida to have something like that in place. And I think just what everyone just reported out on, it's really obvious that there's a definite research need. And, it's really wonderful to hear all the great work that's being done. And I look forward to watching it bloom in Florida as well. Leslie Sturmer is on the call too. She might have some input for the whole group as well as related to that.
Leslie, you're welcome to speak up if you want to contribute to the conversation.
Well, I don't really have much more to add than what Angela said, is that, yeah, we are seeking to look to see what the policy and legislative requirements may be for the state of Florida to engage in a nutrient trading program. We are looking at a couple of funding resources to be able to bring Tom Ankerson. He is the director of the clinic at the UF Law School, and would be very instrumental in applying his skill sets to addressing that question, since it seems to be a very state-specific set of requirements. And that really didn't come out in Matt's talk. But yeah, so we understand the need to address this and trying to move forward with the resources that we have in the state to address that.
Wonderful. I did get a request from Bill Arnold. I think he wanted to mention a couple of things before we, of course, wrap up for this meeting. Bill, now you have the opportunity to do so. So feel free to go ahead.
Well, thanks, Sonora. Really not much. I was just thinking that one of our research needs might be to build a model that is quantitative and that really takes this from start to finish on, you know, the dynamics and the costs and everything associated with using seed, using adults, using larvae, the cost of production, et cetera, on through planting, you know, where are the bottlenecks all the way through to harvesting or whatever goal it is you're trying to achieve. You know, you should, you know, if you're trying to do nitrogen reduction, you could kind of start backwards with Ashley's illustration of a clam in the sediment, and this is what goes into nitrogen and nitrate and ammonia, et cetera, you know, work back so that you can actually compare scenarios and say, you know, and start drawing predictive conclusions regarding alternative approaches to reaching a goal, whatever that goal may be. I would think that, you know, building a conceptual model at first, which would be relatively easy because it has no numbers associated with it. It's basically just pathways. That's something that could probably be done without funding, but, you know, building a truly quantitative model, I would think would be a very fundable project. But maybe it's already out there. I don't know all the details on this. I see that clam environmental benefit calculator that Angela provided. I don't know what's behind that, but I think that that's something that, you know, that's a research project that should at least potentially be considered.
Yeah, so just real quick, Bill, that I'm glad that you brought that up because I was just thinking that needed to be addressed real quick. It's, you know, it's Leslie's on the call, and this is, you know, a shellfish, University of Florida shellfish program tool, which I know Leslie knows the entire history of. So that is one of the only tools available right now for some of these calculations of the environmental benefits of clams. So I'm gonna let Leslie, is it okay, Denora, if Leslie addresses that real quick?
Absolutely, of course.
Oh, okay. I'm not exactly sure. I'm trying to do two things at one time. It doesn't work very well. So were you, Angela, talking about the environmental benefits calculator we did that's online?
I am, yeah, because that's one of the things that local governments look to when we're quantifying these ecosystem impacts and environmental benefits of claims. That's one of the only tools available for Florida right now. And so I think over the last 3 days.
Yeah, I want to clarify that the values we determined in that, in a project we did, I forget who funded it, that doesn't matter. Shirley Baker did the clearance rates, filtration rates for several sizes of harvest sized clams from pasta to a 1-inch little net. So that data is incorporated in the calculator. Then we only did extraction values for nitrogen and carbon. So again, when you harvest that clam, and we use 3 sizes, what not just the meat itself, but the shell, what was the extractive value of nitrogen and carbon? And those are the values that are used in that calculator. We did not look at denitrification as the rates. We did not use it net rates. And my understanding from Matt, or at least the presentation we heard from, what is that, Blue Oysters, that's handling the nutrient credit program in Maryland or Chesapeake Bay, that's the values that were used, extractive / harvest values, not net values. So that is available. It is the values associated with those services. We had a economist in our Food and Resource Economics department there at UF use replacement values for both nitrogen and carbon, and that's how we came out with the dollar amounts as well. So you plug that in by county, because replacement values will change by county since we're using wastewater treatment plants, and the cost to operate and construct one vary by county. So it's very county specific as well. So you enter your county, the number of clams that you project to harvest or your production over the year, and it calculates that out. Nitrogen removed, carbon, and the value of those services to the farm. It's pretty simplistic, but that's the data we had at hand to generate that.
No, I think that's great. And you know, it's like we've heard, there's so much work to do, but that's how you have to start. You know, you start with what you have available, and you keep on building up. And like we saw from Matt's presentation, there's a lot of things that we need to find out or figure out. And while, yeah, it might be very state specific, some things, there's still the science. I think that it was a great example of him showing us the process of all these things that we need to really figure out before, you know, we can make like the program work or something like that. But it's a good beginning, and we have great progress. And I think it's just a matter of building on that for sure.
So Denora,
yeah, go ahead. But
what I'm thinking of would be a much more complex start to finish model. And one thing that model would allow you to do, even just with estimates of some of the quantitative components, because it agrees there's a lot of things that need to be modeled, might help you to focus on those things that have the most significant impact to achieving your goals, so you can focus effort and your money on those items that will produce the best benefit, the best return on your investment.
Gotcha. Thank you, Bill. And thank you, everyone. And now I'm gonna just open the floor for, since we like went over all the group discussions, and I would like now to welcome anyone who wants to comment anything further, any ideas, any thoughts. Please, you have the floor now. So don't be shy. We're all here in this together. I think Todd might be. Todd, do you want to, were you going to say something? I don't know. I got at this little microphone. Nope. All right. Well, I think this has been a very productive conversation, and I have incredibly enjoyed it. It's been so eye-opening, but also like this overload of great info. So I think for our goals have definitely been met for this workshop, and we look forward to be putting it all together in a summary, and of course sharing it with you guys. Our recordings of the meetings are going to be available as well. So if you miss something, or even if you just want to listen to a portion of the meeting again, because you know there's something else you want to reinforce, or an interesting idea, it will be widely available for everyone. I know that we also, everything that is shared on the chat, we download documents and look for them and put them in a Google Drive folder for each day. That way it's easier to look for something that you remember maybe was shared the day before or the day after. So it will be, all of it will be widely available for sure. And if there's anything specific that we didn't share with you guys that we had or it was spoken about, feel free to reach out to us and ask us for that. Bill, were you going to say something? I see that you don't have your microphone anymore.
I was going to just make a brief comment.
Yeah.
There have been some concerns expressed that we were a little too negative in this workshop, and we didn't mean, I don't think anybody would say that they meant to be negative. We meant to be rigorous and realistic, but you know, but we aren't in any way, or it's way too early to be giving up on these ideas. And I think that Bradley showed that there's a lot of potential here. Nothing's going to be easy, but it is doable. And the fact that we had this workshop is a positive statement. So please take it in that manner. You know, yeah, it's a tough topic, and we're being a little tough ourselves, but only to drive things forward.
Yeah, I think that's a great comment. And sorry, I just muted myself. I definitely think this is a startup of a conversation, and this is just to bridge that communication gap among the different groups, and really take a look at what is it out there. You know, as we suspected from this couple of days of conversations, it has become clear that there's still a lot that, you know, we don't know about the ecological services performed by the clams, and that we certainly need in order to support or validate even further their use for restoration. We have heard from the experts in the presentation that there is also some preliminary research that can provide us an idea of what to expect. And also they laid it out very clear for us that, you know, what are some of the next steps that we need to do, and understanding what are some of the knowledge gaps, and also how clams work as part of the entire ecosystem, and how their restoration might not only impact the sediments and the water quality, but also other trophic groups such as the predators that feed on them, or perhaps other viable species. Certainly restoration efforts using clams, you know, we heard from the many fantastic groups here in Florida, is becoming very popular, and we hope that we have made all the groups aware of each other so that you can communicate, help each other in any questions you might have. One important conclusion I think that we heard is that we have the potential to help our estuaries, and of course, restoration of clams might not or should not be used as the one solution for all of our estuary issues. They can serve as one of the many tools that we need to be used as part of the larger management of water quality strategies, so that we can ensure that our estuaries are healthy. And this has been also, I took as an important message from this meeting, is that we need to really understand the systems that we're working with very well, and also why our estuaries are degraded. It's important and a first complex step, but it's often necessary to understand if they are even in the right condition to sustain clam populations over time. That way we're not putting a lot of effort or spending a lot of money adding clams in maybe particular locations of the estuaries where they might not survive. We hear over and over that is super clear, super important to identify what are your restoration goals. And by first identifying this question, I think then it can help us understand what is the best approach or the process. What, you know, are we trying to restore water quality? Is it less nutrients? Is it seagrass enhancement? So that's also gonna dictate whether are we thinking about removing the clams after some time, or actually leaving there to stabilize the sediment. I did see there is one raised hand. I'm gonna ask Tom McCrudden, do you have a comment to say?
Yes, I've been involved with Florida Oceanographic over on the IRL for over the past 20 years. And we had some good research that we did an oyster deployment inside a containment boom that was successful back when Vincent Encomio was there. And I'd like to try to see if we can get that information out to everyone to show that we did have a successful deployment. And we showed positive recruitment inside that deployment. So there has been positive projects like that we should try to get out and share with other people.
Absolutely, Tom, I agree. I think that's very encouraging. If you have any reports or any on that, please feel free to send it, and we'll make sure that we send it out to all the participants, so everybody can have all the great information out there for sure. We definitely need those those good stories to spread the word for sure. Alrighty, so okay, yeah, I want to point out that this workshop was focused on the restoration aspect using clams, and while a lot of the studies that we heard from our talks were cited from shellfish leases, this is in no way an indication that the aquaculture industry is not doing a good job, or they have been an incredible contribution to, you know, our local economy, supply of food. And what we're really trying to do in this workshop is to point out what we do know and not know about the ecological services provided by clams. And as suspected, we don't know that much, and there's still much work to do. So when we're not saying that there's no actual ecological benefits, we're just not able at this time to quantify them adequately. So hopefully, as a result of this workshop, we will be able to make accurate estimates of the different ecological services or suggest farm management practices that might maximize the potential benefits. And this will hopefully help even further to the farmers as well for another market. I want to finish by encouraging collaborations among the different groups. I will share the list of participants, then you can reach out each other. I want to give out a shout out to our farmer partners who are the ones who are out there every single day, and they are developing a lot of the ecological knowledge that actually help us, you know, do what we know, what we need to know about clams. So thank you, and they always supplied us with the clams that we need. So we definitely need them on board on these efforts. And I see that Angela has a raised hand, so please feel free to go ahead and comment.
Hey, no, I was just going to say the exact same thing. I was just going to thank the farmers that are on the call that took the time out of their work schedules to join the restoration workshop for all 3 days. And also just to remind the group that those farmers are out there and have a lot of that ecological knowledge that's not in the literature, but they have so much insight to give to the community.
Absolutely.
On some of these topics.
They are the ones that every day they're out with the clams and they know them. So they have so much knowledge that we might not even find in the book. So I think working closely with them, and it's really valuable for all of us scientists. So thank you, of course. And we definitely want to continue this going and encouraging that. And with that, I think I want to open anybody else if they have any comments. We will send a post workshop survey, so please do take the time to complete it. This really helps us see you know, comparisons with our initial survey, and also evaluate how we did with this workshop, and hopefully keep, you know, making things better and more available for everyone. So any last remarks from anyone? Speak now. Alrighty, I don't see or hear anyone speaking, or did I miss any comments? I don't think let's see, perhaps post-workshop survey to identify who needs terms of research, contact information. Yep, we will definitely send out a survey, so please it's gonna just take a few minutes to do that. And I think it will be definitely helpful for everyone. And thank you so much all for participating and giving your feedback. We definitely could not have done or gathered this much information without you guys, and all the speakers for taking the time. So thank you so much, and thank you for your support for sure.